Hello friends, welcome to SR Academy.
Welcome to SR Academy. Empowering minds, igniting curiosity. Your gateway to knowledge unleashed. Let's get started.
In kidney, like for all these mechanisms, you need cells to be active. If the cells are not active, then that particular mechanism will not occur efficiently. If that particular mechanism doesn't occur efficiently, then you will be in trouble. Then you will definitely exposed to the different diseases and inflammations. All those things will be there. So we have to get the cells from these particular healthy cells, from these particular organs. Use the tissue engineering, grow them in the scaffold or a biomaterial or a extracellular matrix with the help of growth factors, and then implant them back into the organs so that they will replace the non-functional cells with the functional cells. So once these functional cells comes, then they will start producing the hormones, they will start producing the nephrons, they will start doing the functions properly. That's how we are going to treat. So it can be a lot of defects will be there. So first we need to see what are the defects, and then how the tissue engineering can overcome these defects by incorporating the functional cells or tissues with the help of tissue engineering. So that way this whole concept will be revolving around these particular aspects. So I hope that you understood the introduction. Let's see the topic. So can we use cells as therapeutic agents to restore the tissue function? That means cells we will be using as a therapeutic agent. That means here cells will act as the treating agents or treatment. Okay, because you are using the cells. Imagine a liver is not doing its function because of the lot of cells are not available. They are not doing their functions because of some infection in the liver or because of some damage in the liver. So now I will use the cells, the healthy cells of the liver. I will clone them, I will seed them in the extracellular matrix, that is scaffold. I will use the growth factors. I will develop the tissue, and that tissue I am going to incorporate in the liver so that the liver will start functioning properly. Understood? So here what cells are doing? They are actually used for treating the liver now. So that's why we call it as a therapeutic agents. So can we use cells as therapeutic agents to restore the tissue function? Answer will be yes. So yes, we can use. So how? The cells are building blocks of the tissues, right? So which contribute the respective functions. If any functions are lacking, that means the tissues are not working, that means the cells are not functioning. So you replace the non-functional cells with the cells, the functional cells, so the tissues will work. So definitely, so respective function will be restored. So cells based therapies are a distinct class where cells are utilized as therapeutic agents to treat pathological conditions. To treat pathological conditions, cells can be used as a therapeutic agents. So however, cell based therapies are not new, right? Definitely it is not a part of new tissue engineering or kind of stuff. It has been used very widely from the beginning itself. So from a lot of the if you go trace back even in the later 90th century and all, it has been used. So however, the cells therapies are not new. Because of many decades, people are practicing blood transfusion. See, blood transfusion is one of the example. Why we use blood transfusion? Obviously, here we will be using the blood cells like RBC, WBC, and all transfusion to anemic patients to restore the oxygen supply. To make sure that in order to get the oxygen supply, we will be using the blood transfusion. That is one of the practiced methods since many decades. So similarly, platelet transfusion is also there for clotting defects, blood clotting defects. Wherever the blood clotting defects is there, so there the platelet counts will be less. So we are implanting or we are transfusing the platelets or incorporating the platelets in order to get the property, that is blood clotting property, so that we can avoid the blood clotting defects. Next we are having the bone marrow transplantation. That is also cell-based therapy, which is practiced in decades ago. So for cancer patients, the bone marrow transplantation is used, or practiced widely. So these are the three major techniques which I pointed out for where the cells can be used as the therapeutic agents in since decades. Blood transfusion is there, platelets transfusion is there, bone marrow transplantation is there. So few examples. So cell-based therapies can also be used to repair the cartilages. So for cartilage also, to restore the function of the cartilage also, we can use the cell-based therapy. Next, we are having hepatocytes and kidney cells are also used to carry out the function of livers and kidney respectively. Definitely the if the liver is not functioning, if any liver defect is there, then hepatocytes are not working properly. So replace the hepatocytes, incorporate the hepatocytes so that you will get the healthy liver. Similarly goes with the kidney cells for kidney. So similarly we are having beta islet cells for diabetes, sheets of fibroblasts for ulcers and burns, genetically modified myocytes for the muscular dystrophy that have been used clinically with varying degrees of success in past few decades. Okay. So cells and their uses. So source of the cell is very important. First you need to identify the source of the cells, whether it is autologous, or that means whether we are taking it from the same person or a different person or a different species. All those things plays a very important roles. Okay, so how do we use the cells as therapeutic agents for various defects? So that plays a very important role. This whole topic we will be using that only, we will be explaining that only. So we can say that the cells can be used as therapeutic agents to restore the functions, okay, wherever there are defects are there. So there are 5 defects we are going to discuss: mechanical defects, metabolic defects, synthetic defects, communication defects, combination defects. Wherever the mechanical defects are there, those whatever the responsible cells are there, you introduce the healthy cells so that the mechanical defects can be treated. So same thing goes for the metabolic defects, synthetic, communication and combination defects. So source of the cells. So there are 4 different sources of the cells are there. That means the source of the cell is nothing but it's a fundamental question, from where exactly you are going to get the cell. Okay, so autologous from the same person, allogenic from the different person, syngenic again from the different person of this thing, xenogenic from the different species. So this is how it is in the picturized way we have detected. If this is a mouse, if I am taking the cells and I am introducing back to the cell, same mice, for example here some particular organ is having some issues, so but half of the organ is having healthy cells. Now I am taking the healthy cells and using the tissue engineering and preparing the tissue and then incorporating or implanting back into the mouse A, so that I will get the particular same cells. So that is autologous. So immune rejection will not be there, immune system will not see them as a foreign cells. So next we are having the syngenic. Syngenic is nothing but with a same person, like, or you can say same family person or something like that. So you can use that syngenic from here to here, from different person. Okay, that will be allogenic. So that here more in immune rejection, that these things everything will be there. And then xenogenic, the whole different species will be there. Here we are having mouse, here the rat. That will be a little bit difference in this particular what you say, genetically there will be difference. So that's why we are having the xenogenic, where the species will be different, so that you will experience the what you say, the immune system will be active. It will start and producing any inflammation or infection to the particular or a damage to the particular cells which have been incorporated. For that, for xenogenic we will always have to go for xenogenic, allogenic and syngenic. For all these 3 things we will have to go for immunosuppressors. In some cases syngenic doesn't require immunosuppressors, but allogenic and xenogenic, for if you are taking these cells from here to here or here to here, you have to use the immunosuppressors, so that the immune system should not produce antibodies which should not come and kill the healthy cells which is taken from the different persons or rats or different species. How do we use cells as therapeutic agent for various tissue defects? So answer is, to answer this question, we should first know what are the different types of the tissue defects based on the functions. So we know that mechanical, metabolic, synthetic, communication, combination defects. That means this is definitely is classified based on its functions. Mechanical function, we are having all these particular bone, cartilage, all those kind of stuff. Whereas when you are talking about metabolic, it should be the production of the hormones, certain proteins, or certain biological compounds. Then the same thing goes for synthetic also. Next, communication, that is for the nervous system. Then combination defects will be there based on this particular skin grafts and all. Next, for example, the major organ is affected by affected in mechanical tissue defects is a cartilage or a bone. So since the major functions of these tissues are providing the mechanical strength to the body, so if any damage happens, then we will experience a lot of pain in the knees because the cartilage is not working properly, because function of the cartilage is to give the cushioning, and if that cushioning is not there, then your two bones will start a lot of frictions will be there in between the two bones in joints, so that will start explaining the heavy pain and all. So now these defects can be treated with the cells which are healthy cells, so that they will activate this particular function of cartilage, so that the cartilage will do its function, it will give the cushioning, and then you can that will reduce your pain. So we are having the mechanical, this thing defects, usually we go for the bone or cartilage, because they where exactly the bone and the cartilage plays a very important role in our body, which will give you the mechanical property. If any defects are there in bone and cartilage, that will come under mechanical damage. If any defects are in the liver, then we will be talking about metabolic. And because the whatever the particles, whatever the hormones it secretes and all. And then synthetic, if you are talking about, then it will be definitely in pancreas, because it will be in the production of synthesis of insulin. Next, for communication, we are having this nerve, and combination defects, we are having the defects in the skin. So cells used to restore the one by one we will see. So cells used to restore the mechanical defects. That means a major component of cartilage is what? Extracellular matrix, or you can simply call it as a ECM. So where terminally differentiated chondrocytes will be dispersed at lower densities. In this particular cartilage, the chondrocytes are the cells which will be dispersed at lower densities in the ECM. So this architecture promotes the frictionless surface with pain-free motion, so that if because of this in between joints and all, this particular cartilage will be there. So when the one bone is it also acts as a one bone connection between one bone to the another bone. So in between it will be there. So if the cartilage undergoes any defects, especially when you age, so what happens, that particular cell chondrocytes will not be there, or you will be exposed to the osteoarthritis. So this, because arthritis is because of the deterioration of the cartilage, which is including the loss of chondrocytes. That means the chondrocytes will not be there, enough chondrocytes will not be there to give the significant cushioning, and as well as ECM structure or architecture is also damaged because of the deterioration of cartilage. So because of these things, you will experience the heavy pain in the osteoarthritis. So this deterioration cannot be repaired due to the lack of blood vessels, because we don't have any blood vessels to there. So whatever the medications will be given, that will not reach there, so resulting in acute pain during movement. Okay, whenever you move, there will be acute pain. So cartilage defects in the knee can be treated by using autologous transplantation. That means you are taking the same where the different, for example, if you are having the right leg pain because of the deterioration of the cartilage in the right leg, then we can take the healthy cells of the left leg cartilage, and we are going to use the tissue engineering and clone them or seed them, produce chondrocytes, harvest them, then you implant back into the cartilage, so that they will do the function of cushioning, so that you will be free from your pain. So briefly, a biopsy is collected outside the affected area from the patients. That means any healthy cells can be taken out. then subjected to the enzymatic treatment where chondrocytes will be harvested. That means more chondrocytes will be produced. Now these chondrocytes are subcultured. That means again you are going to subculture it for expansion. That means to get more copies or more number of the chondrocytes, and then inject back into the patients at the lesion, wherever there is a lesion is there. So about 200,000 patients are undergoing this kind of treatment of cell-based therapy every year. So autologous transplantation of the chondrocytes can give a big relief to the pain in between the joints. So this is how autologous transplantation of chondrocytes in osteoarthritis patient works like. This is a normal healthy cartilage. This is how it looks like, and this is the this is osteoarthritis. Arthritis is nothing but here, see, this is how it is deteriorated, or you can see it is almost like damaged. It's because of many reasons, usually in the age problem after 50 or 60 and all, this particular damage will be there. So you take the biopsy of healthy cartilage, whatever the healthy cartilage is there, this pink one, red one is deteriorated. Now whatever the pink one is there, you take that as a for biopsy. That means biopsy is nothing but taking out those particular cells. Now you break down by the enzymes. You use some enzymatic treatments that cells will be break down, and it will be reduced to at cellular level. Now the cultivate these particular cells for 14 days, that is 10-fold increase of cell member. If 10 cells are there, 10 chondrocytes are there, then after 14 days you will be having 100 chondrocytes. That is 10-fold increase in the cell numbers. Now these particular cells, you again go for the enzymatic treatment. Here trypsin treatment is widely used. Once you get that, you will get this particular suspension. Now this particular suspension has got the chondrocytes which are being harvested. Now inject these harvested chondrocytes back into the particular the cartilage. So where the damaged cartilage is there, or you can say where this particular deteriorated cartilage is there. When you introduce here, what happens, the cells will start growing, because once you introduce here, then then finally they will give you the healthy cartilage, normal healthy cartilage. So this thing is reduced, or this thing is completely replaced with the healthy one. So whatever the deteriorated cartilage is there, now that will be the new cartilage. Around 200,000 of patients they are going under this particular kind of treatment every year. So cells based, that was about the mechanical defects. So that's how the tissue engineering can help. Okay, so to cells used to restore the metabolic defects. So metabolic defects here, loss of liver function is mainly by hepatitis C, definitely. So if the liver is not function, that is maybe because of the loss of cells, loss of function, okay, of hepatitis B, or excessive alcoholism, or even cancer. So in any of these three things, what happens, the liver will lose its function. Okay, so the extracorporeal hepatocyte based bioreactors, that means bioreactors in the sense where we are going to cultivate the particular hepatocyte cells. So they are used to culture the human hepatocytes, and that can be able to maintain the liver function in the bowel. Alright, so bioartificial liver. So that this particular bioartificial liver will act as which will be the result of hepatocytes based bioreactor. Why we call as bioreactor? Because we are going to culture the n number of cells, hepatocyte cells, which are responsible for liver functioning. If the liver is losing its function because of any reasons, whatever mentioned about cancer, alcoholism, or hepatitis C, so if you that will be a direct you can say that the hepatocyte cells are losing its function, that's why the liver is losing its function. So if you can culture the human hepatocytes in a bioreactor, okay, then we can treat this particular metabolic defects. Hepatocytes are the chief cell presence in the liver, which are highly proliferative cells. So usually bioreactor with hollow fiber cartridge charged with the immobilized hepatocytes, that is, you can call it as intraluminally or extraluminally, okay, is used. So this is the setup where the bioreactor will be look like this. So this device is connected to the outside the body. It will be connected to outside the body to the blood or a plasma circulation of patients, but it will be connected to the blood or plasma circulations. Now the hepatocytes can also be embedded in the collagen hydrogel, okay, to avoid hydrodynamic damage, and then injected in back into the hollow fibers. So due to the hydrogel nature of the collagen, that is a polymer nature of the collagen, so it contracts, thus forming a lumen. So it will form the lumen, so which helps in the diffusion of the medium and as well as plasma. So the diffusion is occurring that. So that diffusion is occurring mainly because of the lumen which is being formed. Okay, so this type of extracorporeal device, that means which is kept from the patient out of the patients, that can improve the detoxification and synthetic and as well as even regulatory function of the diseased liver. All these particular things, issues of the liver, that can be regained with this particular approach. So that was about the liver. Okay, metabolic defects. Next we are having the cells used to restore the synthetic defects. So we know that insulin is produced from the pancreas. In insulin what happens, insulin dependent diabetic patient, what happens, pancreas loses its ability to secrete the insulin. So if the person is a diabetic patient, it means that his pancreas is not able to secrete the insulin properly. So allogenic cells, that is taken from the same person, are used, that is the cells isolated from the same from the thin pancreas, and then injected into the liver portal vein, thus enabling the pancreas to secrete the insulin. Okay, so make sure that you are giving the healthy cells, so that the healthy cells will replace the non-functional cells, thus the insulin synthesis will begin. So we can treat the diabetes in this manner. So however, since the source of the islet cells is allogenic, there will be immediate rejection due to the reoccurring autoimmunity and as well as inflammation. So even in allogenic, so sorry, this is allogenic in the sense we are taking from the other person. So definitely there will be immediate rejection, because the immune system will take it as a foreign body, because allogenic from the different person. So recurring autoimmunity will be there. Inflammation also can be possible. Hence, physical separation of the donor cells from the immune system is mandatory to avoid the immune rejection. That is very important here. So in order to avoid, we will use immunosuppressants, that this and all, that is different. So here, since we are talking about physical separations also, that is also can be done with some membrane. Okay, some membrane should be put so that physically they are not coming in contact, so that if they come into contact, the immune system will be activated and they will produce the antibodies, and they will come and damage these donor cells. That should be avoided, okay, that by using physical separation here. So this could be achieved by using, how we are going to do the physical separation, by using encapsulation of the donor cells using semi-permeable membranes. Because why semi-permeable membranes? Because we need to have to synthesize insulin, we need to have some particular nutrients that should be passing through the membrane. So which is selectively allows the passage of particular nutrients and insulin, while preventing the passage of the antibodies and the and biomolecules, other biomolecules. So that even though the immune system has been produced antibodies, this particular membrane will not allow those antibodies to pass through its barrier. So it will allow only the nutrients and insulin. So that could damage whatever the biomolecules are there or antibodies which are going to damage the implanted cells, that it is going to restrict. This is how it looks like. So this is macros capsules for the encapsulation of the islets of cells. We have glucose, which is a nutrient. We have insulin, which is a nutrient that will be permeable, that means that can go inside this particular thing, the membrane, semi-permeable membrane, but the other things like biomolecules, antibodies cannot go inside this. Okay, so that was about synthetic defects. Now speaking about cells used to restore the communication defects. So communication defects, in the sense, you know that it's about the neurons or nervous system. So nerve injury creates a gap or a cavity due to the loss of the myelin sheath. so thereby loss of impulse transmission. That means if there is no myelin, no image is there. Okay, so just imagine, you might have studied about how exactly the communication happens. Communication happens when one nerve is coming into contact with the another nerve. Okay, and the linkage will be in synaptic linkage, right? Then dendrites will be there, the linkage will be there. There exactly what happens, the impulse or electric impulse will be transferred from one neuron to another neuron. That's how the communication happens in the nervous system, right? So even in brain also. So this, if the nerve is injured, what happens? It will create a gap or a cavity due to the loss of myelin sheath. Maybe because of the loss of myelin sheath. If the myelin sheath is lost, then there is no, it will create a gap. Then there will not be a connection between one nerve to another nerve. So thereby a loss of impulse transmission. So now we need to regenerate the nerve. So regenerating the nerve, especially in the central nervous system, is really challenging, since the native environment axons in the central nervous system do not regenerate substantially due to the surrounding inhibitory environment. Whenever these axons wants to regenerate, what happens? The surrounding environment is such a way that it inhibits that regeneration. So that is one of the major challenge in the central nervous system. Hence the major aims of the cellular therapeutic inventions in the restoration of the communication defects are to bridge the cavities. That means make sure that there is a bridge between whatever the cavities are there, and then to replace the dead cells, whatever the dead nerve cells are there, remove them and replace them with living cells, and then to create a permissible environment for axon regeneration. So make sure that whatever the inhibitory environment is there, no, for axon regeneration, which is inhibiting the axon regeneration, make sure that the environment is good for axon regeneration. So in three ways we can treat these particular communication defects: to bridge the cavities, to replace the dead cells, and to create the permissible environment for the axon regeneration. So cells such as Schwann cells, olfactory and shielding cells, that can be used to achieve this particular above three goals. Now the advantage of using Schwann cells is that it mainly consists of, or it is mainly because of it can produce ECM, cell adhesion molecules, and neutrotrophins. These three plays a very important role that for peripheral axon formation. If these ECM we are having, if the cell adhesion molecules are there, if neutrotrophins are there, then nothing can block or nothing can stop axon reformation. So hence this provides a permissive environment for the axonal growth. Axons will start growing. So that is how we are going to treat the communication defects. You have the damaged nerve, replace it with the three different ways. With the Schwann cells, we can get the ECM, cell adhesion molecules, and neutrotrophils. With that, we can create the environment which can be which allows the axonal growth properly. So next we are having the cells used to restore the combination defects. So we are having the combination defects especially in the skins, right? So variety of skin ulcers, burns that are treated by using transplantation of fibroblasts and keratinocytes with no immune rejections. So this therapy is almost well developed and lots of clinical products are now available commercially. Okay, which is a very common technique. Okay. Next classic example is the burn injury. You take the burn injury that is classified as first, second, and third degree. First is when the epidermis layer is burnt, the second is when the endodermis layer is burnt, and the third degree is when the the lower layer of the skins, everything is almost burned. Okay, that will be a very severe injury. So the first degree burn, what happens? Epidermis alone is injured, though it is painful, it can heal very rapidly and does not require too much medical attention. So usually when you touch some hot devices, or you can say any instruments, or which is too hot and all, then suddenly you will get that kind of reddish or black kind of stuff will be there. So that means that is only epidermis has been burnt. Okay, that is the first degree of burn. So eventually after 1 week or 2 weeks you will get the new cells or new layer of the skin, right? So that everyone might have experienced. The second degree burns damage both layers of the epidermis and the dermis. So which is epidermis. So second degree the epidermis, and the third one the dermis, while the third degree burns destroys full depth and also underlying tissues. Whatever the underlying tissues are, that will be gone. So that will be a severe injury. So during the skin grafting procedure, what happens? Skin is taken from the different region. You take the skin from different region of any part of the body, meshed and placed at the site of the injury. So this helps in the regeneration of the affected skin, because you are taking it from the same person. There is no much problems. Though skin grafting is widely used, secondary surgery is required to graft the skin and risk of the infection at the site of the injury. Even though if you use the skin grafting, the secondary surgery should be done in order to avoid the infection at the site of the injury, wherever the injury is there. So in addition to that, the patient has to pay a lot of cost associated with this, particularly involved in the secondary procedure, surgery, all these things and all. Thus there is a requirement to develop some synthetic materials which could be used to graft grafts to regenerate skin, so that we can avoid the surgery and associated cost with it.